All right, okay, I'll play something random.
All right. Lads on tour. It's the lads on tour. There's lads in there on tour.
There you go. That's all you're getting out of me.
Okay. So, welcome to Lads on Tour. I am Stars of COVID, a software engineer who has spent a lot of the last 2 years looking at COVID stuff, but definitely not an expert and wouldn't pretend to be one. And with yes, we can hear you, Graham. Fantastic.
Oh, fantastic. Sorry about that.
With it's all right. You're here now. With us this evening is with me this evening are my co-hosts Brent Lee and Dr. Ivor, and our special guest this evening is Dr. Graham Bottley, who is logged in as Swaledale Mutton.
Yes, I am indeed. My alter ego.
So, Graham, why don't you tell us a little bit about yourself and why would I listen to a sheep farmer about COVID?
That is a very common question, as anyone who follows me on Twitter will know. I've been a research scientist for 27 years, something like that now. I did a degree in biological sciences, a PhD in immunology. I've been a postdoc immunologist. But about 17 years ago, I decided to get some sheep. I'd always wanted sheep. And I thought, why not? So I got some sheep. And for about, as I say, nearly 20 years, I've been supplying mutton and so on. And I now work for myself. I no longer work for a university. I work for myself and I train research scientists. And I train them in flow cytometry, which is a research technique that's commonly used in immunology. And I train researchers in the UK, in the US, and I'm involved in a lot of active research projects. So I help people set their experiments up, analyze their data. I get involved on papers and so on. And I do have 2 Twitter accounts. My other one is at Incytometry. But I tend to respond to whatever I see with whatever account is open, hence the confusion that sometimes occurs.
I have to admit that the confusion that sometimes occurs gives me life. So I know you keep a spreadsheet. We will get into the serious stuff. We know you keep a spreadsheet of your most exciting confusing. So what's the big one on your spreadsheet, the most amusing one?
I think the first one, interestingly, because what's interesting is very often on the Incytometry one, it says in my bio, immunologist, and very often someone says, oh, that doesn't happen. You say, yeah, it does. And I think they see the immunologist and they stop responding. They stop getting involved with it. Whereas with the Swaledale Mutton one, there is this, you're just a stupid sheep farmer. What do you know? And so they tend to push their luck. They tend to push their half knowledge. And I think the very first one really that happened was Simon Dolan's sister. So he was the one involved in the big legal case to try and stop lockdowns and so on, whilst at the same time, I think, if I remember rightly, selling masks. And so on. And his sister was on Twitter, and I didn't know at first when who she was. And for about 3 days, we went backwards and forwards, and she was trying to come up with various bits of science that was absolute nonsense. And I kept proving her wrong again and again and again. And eventually she said, you know, what gives you the right to make all of these scientific claims? And I just said, well, I'm a PhD immunologist. I've been working in this field for 27 years. I've got 70 plus papers, and I'm working on this project. And all of a sudden, she I don't think she posts anything. I think she blocked me almost immediately. But the next thing you know, there were about 100 responses going, this is perfect, and the mic drop one, and the boom, and all of the other memes, you know, that and I think that was a good one because it went on for about 3 days, backwards and forwards.
Yes.
You know, and there have been a lot, and there have been some who you know, you get a lot of people saying you really ought to do your research. And I had one person say the other day, I'm not going to converse with someone who doesn't even know how vaccines work, and that sort of thing. And it does make me laugh. And people do say, why do you still engage from that account? Wouldn't it be better to put scientists as well in your bio? But actually, as I was saying, I think people engage more and they're drawn in. And what I'm trying to do is I'm trying to debunk information. And if someone makes a claim and then immediately refuses to engage, I think. Oh, hello. Sorry, can you?
Yeah, we can hear you.
My tablet just suddenly went off. I think if people make a claim and refuse to engage anymore, it doesn't show their claim up as completely false and in fact laughable. And I think sometimes the fact that they are shown up, you know, in that way, it's probably slightly cruel. But on the other hand, what it does do is it says to everyone else who is watching that Twitter thread develop that actually they're making an idiot of themselves and their claims have no basis. And I think then that achieves the goal of debunking.
Yes. Okay, great. So I think what we'll do is we'll have a little bit of background about how do vaccines work, how do trials work, and then we'll get into. I have a list in front of me of some of the more exciting claims about the vaccines. I'm not going to go full into like what if there's a 5G chip in it, because that's so stupid that it.
To be honest, you need about a 3-day podcast if you were going to do all of them.
Well, this is true.
Yeah, I mean, well, I wrote a book with 500 of them in it. So.
Yeah, and that's out of date now. You know, the day you released it, it was out of date because new ones come along.
Yeah. So
Vaccines Cause AIDS has come out since. Yeah. Or
ridiculous.
Come back in the headlines again. Yeah.
So let's say there's a new virus out called cooties.
Yeah.
And
okay.
We need to vaccinate people against cooties. How do we go about designing the vaccine and getting it through trial and getting our cooties vaccine to trial or to the market?
Well, what I will say is it's a lot easier now, and by now I mean the last year or so, than it would have been, say, 5 years ago. Because the first step is to find out what you can about it. So find out what cells does it infect, what physiological effect does it have, how does it bind to cells? Because it could be that it binds to a certain receptor that we can block really, really easily. And actually, we've got a drug already on the market that's dirt cheap that works.
Yes.
So first,
not ivermectin then.
Not ivermectin, because that doesn't work. And even if you did have that information, people had come along and claim it didn't work or it did work, or drinking your own urine works or whatever. But the first thing to do is to find out about it, find out what it does, how it binds, what sort of virus. Is it a DNA virus, an RNA virus? What proteins does it have? And one thing that always amazes me when people say, we don't know about this virus. We know a lot about this virus. If I bring up PubMed, which is the search engine for research papers, and these are published research papers, these are not preprints, these are published peer-reviewed papers, and I put just in COVID, it brings up 227,050 papers. Now, some of those are going to be public health, some of those are going to be psychology, some of them are going to be reviews or whatever. But even if half of them are biological research papers, that's 100,000 research papers have been generated in about 2 years.
Yeah.
You compare that with, say, HIV, and if I type that one in, there are 392,000 about HIV, but that is going back to the 1980s.
Yes.
So nearly 40 years worth of research has produced twice the papers than we've got on COVID. So you can find out a lot very, very quickly. And once you've identified the proteins that make up the virus and how it replicates, nowadays with the mRNA vaccines, you can basically sit down and in an afternoon you can design a vaccine.
I don't know if this is even a fair analogy or not, but to me, the mRNA vaccines is almost like programming. You just sit down and you write a description of what you want it to be, and done.
That is a good analogy, because if you know the sequence, and also bear in mind that when I started in research, the Human Genome Project was this big ongoing project, and the goal was to sequence the entire Human Genome Project from one person, and it took years. Whereas I was talking with my wife this afternoon about a project she's involved in, and they do whole genome sequencing on 30,000 different sets of sample from one patient to look for micro changes. So nowadays you can identify these sequences literally in a day. You can identify the bit that where you want your antibodies to bind, and you then have to just find the epitope, the part of the protein you want to bind your antibodies to. Once you've got that, you identify the right genetic sequence. And for someone who knows what they're doing, for someone who works in this field, this is a routine thing. This is like a car mechanic changing an exhaust. Once you've gone in and you've seen what the model of the car is, and you know what exhaust to order, and you make sure you've got the right length and the right silencer box on it and the right spec catalytic converter, it's a half an hour, 45-minute job. And once you've designed it, actually synthesizing the RNA is also ridiculously easy. You can literally switch the synthesizer on. When you come into the lab the following morning, you've got an mRNA vaccine.
My goodness.
And the design bit is literally a day. Yeah, but then you've got to do your trials.
Yes, so let's talk about the trials.
Yes. So the first thing you would do, obviously, you'd synthesize it. And if this was a really urgent disease, so it's killing people left, right, and center, you would put it into lab animals the following day. So you'd synthesize it in the morning. By the afternoon, you vaccinated animals. And then you would be taking blood from those animals at day 1, day 2, day 3. By the time you get to about day 14, you know if your vaccine works in animals.
Yeah.
So 2 weeks after you've designed your vaccine, you know if it works or not. And because there are genetically modified animals that mimic human immunity to some extent, you've got a very good idea if it will work in people. And then what you can do is you can do your clinical trials. So again, what you would first do is you would first inject it into people, and you would check for safety. You would check for side effects. Obviously, you get a good handle on that from your animal tests, but you don't want to be injecting a vaccine that does cause problems.
No.
And if you inject it into the animals and there's no serious side effects, you inject it into a few humans. It might be 5 or 6 or 7. You check them for side effects. You follow it up for a few weeks. You would check. If they all tolerate it incredibly well and they produce a really good, strong human response, you can then roll it out to a bigger audience. And one of the things I come across a lot on Twitter is they say, how can we know what it's going to do 6 months down the line? Or how can we know what effect it has? And I think what people forget is that there are hundreds of vaccines used around the world, and there are millions and millions and millions of research papers that have looked at how the vaccines work at a molecular level, at a cellular level, at a physiological level. And so, again, like programming, if you put a certain line of code in, you've got a very good idea what it will do. You may not know every interaction straight away, but you've got a good idea what it will do to the program.
Yes.
And so if we inject a vaccine and we see a really strong inflammatory cytokine response and a formation of germinal centers, and I'm going very technical here on purpose, and we see all of these certain things happening that happen in every the vaccine, we know that the vaccine is working. And if we then see the Tregs kick in and it downregulates the response and we get the formation of memories, we know that the vaccine is doing exactly what every other vaccine does. And we can then have a really, really, really good idea of how effective and how safe it is. And of course, then you roll it out to the big trial with 30, 40, 50,000 people to test it large scale. And so that's why the whole testing process takes about 6 months for all of these clinical trials.
Okay. Thanks very much for that. It's really interesting how much, how far you can get really early on. I didn't realize that it was, you could sit down one morning and, synthesize your virus and have it in, I guess, mice and beagles and stuff within a day or two.
And this, to be honest, this amazes me as well, because I remember designing mRNA primers, so for real-time PCR, back when I was doing my PhD, or for PCR, and I could design them in a few hours, but then you have to send them off to a company, and you literally had to post off, because the internet was even then the internet was in its early stages. You had to post off your form, and then 2.5 weeks later your tubes would arrive back with your synthesized primers in them. But now these instruments are in most labs. So if you want to do the PCR assays and you want to design new primers, you sit down at your computer, you work it out, and you look at all sorts of technical issues like hairpins and does it bend back on itself and what structure does the RNA have. Once you're happy with it, you put the program into the sequence into your instrument and come back a couple of hours later and you've got a tube of RNA there.
Yeah, I mean, again, this is very like the. I keep coming back to what it's like in the computer science world, but this is like the phone in my pocket is so much more powerful than the computer that I did my final year project on. And if I'd been at university 10 years earlier, I probably wouldn't have had a computer. I probably would have had to go to labs to do work.
Absolutely,
it's a similar process. Okay, so we're going to start with the.
Think of how far it came in terms of vaccine development, where in the past, like the first vaccine, we just had to hope there was a similar virus out there we could inject people with to give immunity to another one. Yeah.
Well, it's literally scrapings from a diseased cow, wasn't it? It was the first vaccine.
Yeah, it was. And we're, in a way, we're lucky that worked, that cowpox actually provided such a good response against smallpox, because if it had been only very slightly cross-protective, if it had only produced a really low protection, there's every chance that boy he tested it on would have died, at which point the whole history of vaccination would have stalled potentially for decades, or even 100 years or more.
Wow, yeah. Okay, so we're going to go on then to, in my head, I put down we're going from least silly to silliest, roughly. Okay, so least silly. I am the #1 tennis player in the world. I am Superman. I am 30 years old, skinny and fit, and will probably be able to tank COVID without a problem. And according to this weird test I got, I definitely had it before.
Yeah.
Why would I? By all means, if you're 50 and diabetic, you should get it. But why should I, as a tennis champion from Serbia, get the vaccine?
See, that's actually a really easy one to answer. And so being able to run a marathon, being able to lift 200 kilograms, being able to play professional tennis means you are fit. It's cardiovascular fitness. Your heart pumps well, your muscles are toned, you've got the right body fat. None of that has the slightest implications for your immune system whatsoever. So you could be a heavily overweight 90-year-old who's never done a day's exercise in their life and have really, really good immune cells. They're not linked in any way, shape, or form. To use a different analogy, it's like saying, I've got a Ferrari sports car. It can do 0 to 60 in 3 seconds and top speed of 190 mile an hour. Why do I need a digital radio? Well, if you're driving through an area with poor reception, you're going to get a terrible signal on your radio. It's nothing to do with the speed. It's the radio. The immune system is completely separate from bodily fitness. So that is complete nonsense. There's nothing in that whatsoever.
Okay.
As for the I've had it before, that's a little bit more complicated. Did you want me to go into the details now with that? Because that's a really common one.
Well, let's at least go into statistics for let's try and get an answer to it.
Okay.
And I know that you probably have the ability to do a 4,000 word paper on it.
Yeah. Well,
and I respect that.
I'll break it down to about a minute or so.
Okay.
Basically, when your body sees a whole virus, you're infected, it makes 20 different types of antibody. But only one part of the virus is actually worth making an antibody against. That's on the outside. That's the spike protein. Any of the other viral proteins, antibodies cannot bind to because they're just not there to bind to it. Okay. So it's like, again, use a good analogy here. If you make an antibody against a car, if you want to stick if you want to get an antibody that binds to a car, you make it against the outer body. You don't make it against the seat or the engine or the robot that made it in the factory, because there's no point having something against the robot in the factory. So you want it against the car. But the way the body works is these are randomly selected. It picks 20 random ones. So it's like having a pack of 500 cards, and some of them are the body shell and some of them are the seats and some of them the engine, some of the robot in the factory. You deal 20 cards out, they're the antibodies you're going to make. And so if you're unlucky and you get one card that says you're going to make it against the spike protein of the virus, only 1/20th of your response is against will neutralize the virus.
So
if you're really lucky,
things.
all 20 might be, but it's going to be somewhere between 0 and 20, and you don't know what that is, and you've really got very little way of finding out. However, if you have the vaccine, all 20 of your cards will be again. Now you've got a pack of 40 cards that you deal 20 from, but all 40 cards are different bits of the spike protein on the outside. So all 20 will neutralize that virus.
Because the vaccine is only producing the bits that are useful to attack.
Yeah. And if you've had it already and you have the vaccine, then what the vaccine does is it switches a lot of those non-protective ones around to protective ones. So even if you've had it, having the vaccine still boosts that protection and brings you up to a fully protected level.
Okay, I think that's a brilliant answer, and I think that it's very clear that, well, to me anyway, it's very clear even just looking at the numbers that there's very, very few people who, well, I think anybody who's medically capable of taking the vaccines should get.
Yeah,
I don't think there's much debate about that.
No, that's, without a doubt. And I did even see a GP the other day say, why would we vaccinate healthy children? And the obvious answer to that is, well, to keep them healthy. I think children need to be vaccinated. I think adults need to be vaccinated. We all need to be, because none of us are safe from it. None of us are safe from long COVID or downstream effects. And none of us, even if we've had it, are protected until you've had 3 vaccines, and that's the best protection you can get.
Can you explain to me then? Obviously, we got the trial results from the Pfizer, the Moderna, and the AstraZeneca vaccine, and we had these efficacy results against infection that were in the 90s.
Yeah.
But as things have went on, we're getting, still getting good efficacy against hospitalizations and against deaths. which is brilliant. But we have this waning immunity against infection.
Yes.
What's going on?
So there's several things going on there. The first thing that's happening is that when you have your vaccine in the next couple of weeks, and when you have a booster in the next couple of weeks, your antibody titers, the amount of antibody sitting in your blood, goes up very sharply.
Yes.
And so your blood is flooded with antibodies ready to fight the infection. Now, what then happens is the body says, well, hang on a minute, we're not seeing it at the moment, and it gradually tones that down. It produces less and less and less till you get back down to background levels. So if you encounter the virus 3 months after your last booster, your antibody levels have dropped quite a lot.
Yes.
And what you've got to remember is this is not an all or nothing situation, because if I were to do an experiment on you and to put you in a room and to spray 100 virus particles up your nostrils, you're probably not going to get infected. If you've never seen the virus before, you may well do. If you've had one dose, you probably won't, because there's enough antibody to neutralize those 100. If I spray 1,000 molecules up there and you've been vaccinated, you're still probably okay. But if I spray 10,000 or 1 million or 10 million, then even if I spray 10 million up there, you could have had the booster a week ago and your antibody levels could be tip top. But so much virus comes in that it overwhelms the antibody that is sitting there waiting for it and infects some cells. Now, once it infects the cells, the antibody can't do anything. Then you rely on the rest of your immune cells, your killer T cells and so on. They then have to kick in to clear the infection that's happened. And that takes time because the virus replicates very, very fast. So with the Omicron one, the one we've got now, that is more infectious. It spreads quicker. And the reason it spreads quicker is because it binds better to the cells that it infects. So a virus that doesn't spread very quickly, that it binds quite slowly to cells. It gets inside quite slowly. It's all quite, pedestrian, which means that you don't need a lot of immunity to stop infection.
So wild type was much slower than Omicron
it was slower. Yeah, exactly. Whereas the Omicron one, it gets in. And so, if you want to imagine in your mind, the Delta one, it gets into your nose, or sorry, the original one, it gets into your nose and it wanders along looking for a cell to bind to. Is this one? Oh, I suppose we might do. Whereas Omicron gets in, bam, it's infected. No mess around, it's in, it's straight in. And so the response to it, you've got to have a much stronger response. And so, but what you've also got to remember with the current figures that we look at the moment, we're seeing more infections. But part of that is going to be behavioral as well, because when we first started vaccinating people, obviously the vaccines were fresh, so we had high levels of antibodies, but we had mask mandates on public transport and in shops. We had limits on the number of people who could mix. We had self-isolation. We had work from home. The stations were empty, the people weren't mixing at work. So we saw a lot less infections then because the vaccines were fresh and not much was being spread around. And when people were exposed to it, they were exposed to small doses, which their high level of antibodies could deal with. Now what we've got quite often 3-4 months since the vaccine for many people, we've got a virus that binds very, very quickly. And people are just mingling left, right, and centre because the messaging from the government is it's all over, do what you want.
Yes. Lick each other and stuff.
Yeah, but more or less, yeah. And it's quite worrying. And I was discussing with some other scientists earlier that the whole messaging is basically, it's over. We don't have to worry about this anymore. It's just like flu. And it's not just like flu.
Yeah, okay. I'm going to, let's see. There's been an awful lot of pseudoscience about the spike protein's so dangerous that you definitely don't want anything in your body that's making spike protein.
Yeah.
And to me, it's a bit weird because if it's dangerous, I'd rather have it in my massive shoulder muscles,
Yes.
where it replicates once, than in my lungs, where it replicates more than once, and has a bunch of other crap with it.
Exactly.
But I'm guessing that there's a more complicated, a more complete answer than that.
As with everything, immunology is complicated, and it is more complicated than that, because a lot of the in vitro, that is to say in the lab, in a flask, in a test tube, a lot of those results that look at how dangerous the spike protein is are using very often what they call pseudovirus. So this is basically an assembly of spike proteins all pointing outwards.
Yes.
It's not quite a virus because it doesn't really have the replication, it doesn't have the genetic material in, but it works quite well. You can label it, you can make it glow, you can look at it down the microscope. So if you want to look at, for example, how well serum from a patient blocks infection, you can add serum and pseudovirus and see how well it gets into cells. And so if you add this pseudovirus to some cells and you show it has an effect, it's not necessarily saying the virus will do the same thing, because they're not quite the same shape. But it also means that the effect you are seeing depends on the context in which that spike is presented. Now, what I mean by that is a single spike binding to a single ACE2 molecule on the cell surface will do something very different to a whole load of spikes binding to a whole load of ACE molecules. Okay, so if you imagine, the catching game you sometimes get for children with like two pads with Velcro on and a ball that sticks to them. So if you imagine the receptor is the Velcro, but the ball is 6 feet across. So one little ball binding to one of these pads does something to a big ball binding to 20 pads, because binding to those 20 pads, you fold the cell round the big ball. Mm-hmm. And that's different to one molecule binding to one receptor on the surface. The cell responds differently. So the cell will do something different if one binds to if loads bind. So one spike on its own, such as you might produce with the mRNA, doesn't do the same as spike in the context of a virus or what they use in the experiments and show damage. That's important. Secondly, when you inject the vaccine, the mRNA is taken up by the cells that surround the injection site. And those start producing spike protein. They don't assemble it into virus because virus proteins are not there. So they can't assemble it into virus, they just make spike. Now, what happens with the vast majority of that spike is the cell produces it and says, well, now what? So if you've got, for example, a pancreatic cell making insulin, there's a little bit stuck onto the end of that insulin that says, export me, chuck me out into the bloodstream. I need to be out there. So there's like a little tag, a little marker, like a little almost delivery label stuck on the end of the insulin saying, push me out of the cell. So pancreatic cell makes the insulin. The machinery within the cell says, aha, this is for export, packages it up and ejects it out the cell. The spike protein doesn't have this export tag, so it makes it. And it produces this protein. It says, well, what do we do with it now? And what it does is what it does with all proteins. It just then, it doesn't know what to do with it. It just chops it up into bits and degrades it. Now, some of it is shown to the immune system, and that's the whole reason we're using the vaccine. Some of it is shown to the immune system to say, this is your enemy, this is what you're looking for. But it's chopped up. It's not exported. It's not chucked out. And there's never been a credible study that has shown detectable spike protein in the blood or the serum from people who have been vaccinated.
Okay, thank you. I think that's pretty thorough. I do have a question from a listener.
Okay.
Can you ask about the Chinese vaccines, or where I can find good information on those in English?
Yeah, I don't know a huge amount about the Chinese vaccine. I believe, though, that the major one is the Sinovac vaccine, which is an inactivated vaccine. So that's basically, they make the virus, they grow the virus in some cells, and then they treat it so that it can no longer replicate. It looks like a virus. It has all of the virus components in, but if it gets into a cell, it can't replicate itself. It can't do anything. Now, these are okay, and these are how a lot of vaccines used to be made: heat-treated bacteria, heat-treated viruses, and so on. And they work okay, but they don't tend to work for viruses, at least, as well as the ones that cause the cells to make the protein that you're looking for. Because if you make the cell you're interested in, if you make your cells produce the protein you're interested in, you're showing it to the immune system as if the cell was infected. The immune system thinks the cell is infected, which means you get the proper response to it. Now, if you just give it the protein, and this can be sort of mashed up virus, not quite mashed up, but you know what I mean. If you show it mashed up virus or heat inactivated virus, you're then relying on some of your other immune cells to eat that virus. So they gobble it up, they chop it up, and they show it to the immune system. And although this works well, and it's how the immune system learns, partly how the immune system learns, you don't get quite the same signals going to the immune system about infection. The body doesn't think it's infected. It just sees something floating around that it doesn't know. So you don't tend to get quite as strong a response as you do to the what they call the endogenous vaccines. And I'm just actually having a look here. So the efficacy in a Brazilian trial for Sinovac showed 50% against infection, 100% against severe disease, and 100% against hospitalization. So
it's worth having then if there's nothing else.
Oh, no, it's there. You know, I mean, these work. These do work. These produce a good response, but they don't tend to be quite as protective against infection as the endogenous ones do. But they all work really well against the hospitalization and the severe disease, because that's a slightly different part of the immune system to the protection against infection.
Okay. So the next thing we're going to talk about then is from your mate Tony Hinton, who I think went skiing rather than talk to you, which was a bit rude.
Yes.
Hashtag Hinton is a coward.
Yes. If I remember correctly, he wanted to speak to a journalist about the finer details of this sort of stuff, and
he did.
You said,
well, I
why
mean,
don't you interview me? Oh.
He basically said Hinton was a COVID expert, and he would debate live on TV one of the BBC's disinformation journalists. And the journalist said, well, that's not what we do, and I'm not an expert anyway. And then Hinton encouraged all of his followers to send sort of abusive tweets and so on. He was a coward. Why wouldn't the journalist debate? No one will debate with Hinton. That's not fair. So I said, well, I will do. And his first comment was, I don't know anything about sheep. And I said, no, that's fine. We'd be debating the COVID vaccine. If you like, I'll respond from my other Twitter account. And the immediate response was a block. Luckily...
Yes, the immediate response was a block.
Immediate response was a block. So luckily, about, I reckon, 200 to 300 different accounts that follow me then tweeted him and said, well, hang on, are you going to debate with someone who is an actual expert in COVID vaccines? To which his response to all of those was a block. And then, as you say, he went skiing. He ran away.
Yeah, he blocked me as well, which was a bit rude.
I'm blocked as well, yeah.
So anyway, like, a thing that he has brought up, and he's correct to bring it up, it is a real thing that does happen with these vaccines, is myocarditis.
Yes.
So is that a worry? Is it not a worry? Is it a worry for some populations of people, some cohorts? What's going on?
The problem with it is we don't know exactly why it's happening. So myocarditis, and again, I'm not a cardiologist or indeed a medic, but as I understand myocarditis, it's an inflammation of some of the heart tissue. And it appears to be related to the immune response to infection. So it's likely that the immune response caused by the vaccine is causing myocarditis in specific people who are susceptible to it. Now, given that we see myocarditis with COVID infection at a very high rate, the likelihood is that the response to the spike protein is causing the myocarditis. Now, my personal feeling is that likely means that someone who is vaccinated and has myocarditis would have got it with the infection as well.
Yes, because it's
their predisposition. Yeah. But the important thing to note, firstly, is that it's very, very, very rare after the vaccine. So I had a look the other day, and there have been 432 verified cases in the UK from something like 40 million doses of vaccine. So that's about 1 in 100,000.
Yes, and that 1 in 100,000 is, my understanding is that it's milder
and
than
it's mild.
The equivalent.
Yes. And the vast majority of those 432 have not even needed treatment. It's not that they haven't, had to go to hospital, and they haven't needed. They've been seen by a doctor, it's been monitored, and it's gone. So they've not needed any medication whatsoever to treat that, and that's the majority of it. And in the vast majority of the remaining ones, it's been moderate and it's cleared up quite quickly. So that really is a non-argument, because if you're avoiding it to avoid the myocarditis, then you've got to hope you don't get infected, because then you're at incredibly high risk of far more severe myocarditis. So it just doesn't hold any water.
Okay, yes, that makes sense.
Can we get ADE in here? Because I think we were going to talk about that one. Yeah,
we'll squeeze in ADE, and then I do have a question, and I'm hoping, Graham. Well, how long does everybody have?
I'm flexible, as long as you like.
Okay, let's talk about ADE, and this was popularized by Poo Lady, I believe. I was supposed to die this winter, and
I've been quote tweeting that. Yeah,
I cashed in my pension. I bought a Ferrari, and now I'm screwed because I didn't die. So don't listen to Poo Lady. Anyway, Graham, ADE.
So ADE is an interesting one. So ADE stands for antibody dependent enhancement.
Yes.
So what it is, sorry, and I had a bit of a brain fade there. It's not like me. And so what this is something that has been seen in both infection and vaccination in the past in rare cases. And what normally happens is, if you have antibodies against, say, a virus, they coat that virus. Certain immune cells can recognize those antibodies. It can see the antibodies themselves, and it can stick to the antibodies, and it can internalize it, and it can eat it. So when I said earlier about you're relying on it eating the virus, that's what I mean. Okay. So what we're relying on is we make the antibodies, it coats the virus, these immune cells eat it to help clear it and further enhance the immune response. Now, if you've got antibodies that don't properly neutralize that virus, they don't inactivate it by binding, then what can happen is all it does is it helps the virus stick to the outside of the cell. Now, if you've not neutralized it properly, and there is, say, still some spike protein being exposed, it can then bind to ACE, the normal receptor for the spike on that cell, and all of a sudden the virus can get into the cell using the ACE. So it uses the non-protective antibodies to get into immune cells easier. Okay, and we have seen it in 1 or 2 infections. However, in order for it to happen, you've got to have three things. You've got to have antibodies that don't neutralize the virus. They don't stop it infecting cells. Secondly, you've got to have a cell that has both the receptor for the antibodies, it's got to be able to see the antibodies, and it has to have the normal viral receptor. It has to have the ACE2. Because if you don't have the ACE2, then it will see the antibodies and it will eat it and digest it. It has to have the ACE2 for it to be able to get in to replicate. And the third thing you need is for the cell that it gets inside to be able to replicate that virus. And not every cell type can replicate the virus. And so with ADE, with this virus, there are no cells that express ACE2 and the receptor for the antibodies and can be productively infected. That is, when the virus gets in, it can make more virus. And there are no cells that can do that. And we've looked at all the cells that can do that, and it's not been found at all. So it can't happen theoretically. Now, furthermore, if it was happening, you would see an increase in cases. It would be obvious in the population data. And it hasn't been seen. And we've given billions and billions and billions of doses worldwide, and there is no signal of ADE, and no lab has ever found it. And it was popularized by the poo lady, and it was also taken up by our old friend Malone.
Oh yeah, he invented the mRNA, didn't he?
So he invented RNA apparently, which has only been around for some billions of years, but he invented it.
Yeah. It was a very strange moment for the world. You have both the Pope and Richard Dawkins whenever that came up.
And so he sort of started running with this ADE is a big problem. And I actually tackled him about it. And I said, well, hang on, you're saying it can happen. So what cells is it infecting? Oh, well, we know that it happens with other vaccines. I said, no. Tell me what cells it's happening with. Tell me the mechanism. Oh, it's a different kind of ADE. I said, how can it be a different kind of ADE? That's what ADE is. That you're infecting these immune cells. No, it's a different kind. I said, well, please explain this different kind to me. Block. So obviously, he couldn't explain this different type of ADE that no immunologist in the world has ever heard of because it doesn't exist. And I think this is part of the misinformation problem. What he's done is he's used his credentials. He said, I am, a really experienced scientist. I invented mRNA, et cetera, trust me. And because he gets big money, and anti-vax is big money. If I chose to be anti-vax, I could be raking it in, but I have morals, I have principles.
Yeah, you'd have to look at yourself in the mirror, and
I couldn't wake up in the morning and look at myself and thinking I'm making money from potentially killing people. I couldn't do that. But for whatever reason he has, and he has people hanging on his every word because of his background. But as soon as he is challenged about the actual science of what he is saying, he blocks or refuses to answer or runs away because he knows that he cannot justify to a scientist the scientific claims he is making.
Okay. I think we also should add in here that he's been banging on about it for 2 years, but there's no peer-reviewed paper what he's produced explaining any of it.
No, there hasn't. And in fact, when people still crop up on my timeline and say, but ADE, and you say it can't happen, they say, what about this? And what they do is they share a paper from 18 months ago that is basically a paper saying, I wonder if ADE could happen, we ought to keep an eye out for it. And you say, well, that doesn't show it. That's a what-if paper. And they share another one that says, ADE has happened in these other vaccines, we ought to keep an eye out for it on this one. And they are the only ADE papers in the entire 237,000. The one saying, what if, maybe, could it, possibly, we don't know. So, no, it's never been seen in the wild. It's never been seen in the lab. One project I'm working on, we did a quick look. No, none at all. None at all. So, until someone proves it can happen, it can't.
Okay, can I do have two people actually who are two listeners. I'm going to ask back to myocarditis. So the question is, if myocarditis is a response to the spike protein, does it make sense to either give somebody a second dose if they had myocarditis after the first dose, or if they suffered myocarditis after the virus itself? Does it make sense to vaccinate that person? Well, I know it's mostly the second dose, isn't it, with the vaccine?
Yeah. Part of the problem is, of course, we don't know the exact mechanism. We're hypothesizing it's the immune response. But what it seems to be is dose independent. So the research that I've seen suggests that if you have had it after you had some mild myocarditis after the first vaccine, obviously the second one is usually given under close supervision, but it doesn't seem to crop up after the second one. Equally, those people who have had it after infection have been vaccinated without it recurring. So if it is a response to, or if it is resulting from the immune response to the vaccine or the virus, it appears to be a specific part of the immune response. And of course, what you've got to remember is when you have the first dose and the second dose, your immune system is doing very, very different things. And similarly, if you're infected and then you have a vaccine, your body does something different. Your immune system does something different each time. You've got different cells involved, different cytokines, different molecules. So having a vaccine, having a booster after vaccination, or having a vaccination after infection will not replicate necessarily the same side effects. And you see it with the other side effects. You see it with things like, the fatigue and the aching arms. They tend to be very different between first and second doses.
Yes.
So, and as far as I'm aware, myocarditis is no different in that respect.
Okay. Thank you. I'm going to ask another call. I'm being very careful not to name anybody, so I don't feel bad whenever we put this onto YouTube. So we've got somebody who has worked in hospitals for months, and they feel like they've been around COVID patients, positive patients. They've been coughed in the face before they had PPE. They've had antibody tests. I assume after that first wave, where surely they were exposed to the thing.
Yeah.
Negative. They want to know if it's possible they got prior immunity from other coronaviruses, if that's really why they don't have specific antibodies, or what's going on there.
Well, there's been no proven cross-protective immunity from other coronaviruses. There are similarities between the viruses, and there have been studies that have shown that there are T cells that respond, that are created from other coronaviruses and respond to this one. That doesn't actually mean you get a protective effect from them. That's one thing to remember. What you see in the lab with these T cell responses does not correlate to what you see in patients necessarily. And we, as far as I'm aware, we've not seen any cross-protective antibody responses. However, what I said earlier about it being very multifactorial, someone who's worked in a hospital with a PPE could well have been exposed to very low doses for a very prolonged period. Now, a very, very low dose may not cause infection, but what it might do is start to build an immune response to it. So if you're with patients, you've got this full PPE and you take your mask off at the end of the day and you get 300 or 400 virus particles in the nose, even with your natural immune responses, your macrophages and your NK cells and neutrophils and things, they could prevent that becoming an active infection, and you start to build a bit of a response. Now, if you then go in the next day and you're infected and you receive another tiny dose, it could almost be like the old immunization. You're receiving a sub-infective dose that nevertheless starts to build an immune response. And so because you're seeing it rapidly and repeatedly, it's almost like you're having boosters. And And well, I mean, the thing is, it's a bit hard to say because this has never really been studied in great detail, and the only way you could realistically do it is almost by taking blood samples, from a healthcare worker on a daily basis during a pandemic. And firstly, it's not their top priority to do that, and secondly, it's a bit harsh taking 10 ml of blood from the same poor nurse or doctor on a daily basis for like 6 weeks. And there are ethics approval and all the rest of it. But, it is entirely likely that repeat low exposure could build quite a strong response. And immune cells do something called somatic hypermutation. So what this is your cells that produce your antibodies, they don't just produce the same antibody for their entire lifespan forever, for years and years and years and years. They start to change slightly. And so they put small changes into the antibodies. And what this means is that rather than hitting the target just in one place, one antibody, that lineage starts to affect the target in 2 or 3 or 4 places. So you broaden your response, you make it more adaptable and also more robust against variants. So it is entirely possible that those people have developed really quite strong immunity. A word of caution though about the antibody test: they look for total amount of antibody. What they don't do is look at how good the antibody is at neutralizing. You could have really quite poor neutralizing, but loads and loads of antibody kicking around. So,
I understand. I think I understand. Okay. Ivor, do you want to ask the next question there?
I don't really have one.
Okay.
What one could be good actually is the sort of that we come out of the European Medicines Agency a month or so ago.
Yeah.
And it was saying that 3 or 4 boosters could undermine your immune system permanently or something like that.
Yes. That bugged me, and I do sort of know someone who works there, and I have actually emailed them to ask what the hell they were thinking, and I haven't had a response yet. That really, really annoys me because there are very, very specific and certain circumstances when immune responses can get reduced by repeatedly prodding it and getting it to work. But they are very, very, very specific. And vaccines are not one of those specific things. If you have a booster, it will enhance your antibody titers. It will enhance all of your immune cells. I'm not aware of a single study in any immunology journal ever, and I have looked, that shows that a vaccine or a booster reduces immunity in any way. Okay, and so I'm involved in active projects, and in those, when we've looked at immune responses after a second booster, after a third booster, we see an expansion of all the cells we want to see expanding. We see production of the cytokines we want to see. We see production of antibodies. Everything expands and gets stronger. And it's really frustrating that an official organization should make a factually incorrect statement. Because the person I know, I emailed and said, what have they based it on? Is this a specific study out yet, or is this internal communication, or what? Why would you say something like that? And I've not heard that. I've not heard back, and I can't for the life of me think why they would say that. But it's very frustrating because immediately that was jumped upon by all of the usual anti-vaxxers saying, oh, look, vaccines destroy your immune system. And it's so frustrating because I know myself they do not do that. I've seen it. So yeah, that drove me round the bend. That did.
Yeah. So something else that probably drove you around the bend was whenever the The vaccine. I can't even say it. I wanted you to ask this one because I can't even say it. The vaccine caused the vaccine mediated AIDS thing that came up based.
Yeah, I do want to bring this one up because this one is sort of like more of a more extreme version of the last claim, isn't it? Really?
Yeah,
it's so dumb.
It is, and actually,
it's sort of like this. The same thing with the sort of spike protein may interfere with the ovaries. Is a less extreme version of the vaccines that are plot to depopulate the Earth by the Illuminati and Bill Gates. Yeah.
So I think what it is actually the last one about the vaccines destroying your immune system. So someone coined the term VAIDS, VAIDS, so vaccine-induced autoimmune disease or whatever it was. So there were several things that all happened at about the same time. So firstly, a couple of researchers wrote to, I think it was the BMJ, and said, we did some work on HIV vaccines years and years and years ago, and one of them we used seemed to increase the infection rate. Rather than decreasing the infection rate of people with HIV, it increased it slightly. And it was based on a virus called adenovirus 5, Ad5. And Ad5 is quite a common respiratory infection. I used to work on Ad5 years ago. And it's quite a common respiratory virus. It's not particularly pathogenic, and it is quite commonly used as a vector for things like vaccines and for gene therapy, because it infects quite a lot of cells quite easily. But it doesn't, even if you had a wild type that could replicate, it doesn't cause much in the way of disease. It causes sort of colds and flu. So they said that they'd worked on this Ad5 vector many, many, many years before, and one of the ones they used slightly increased the rate of HIV. We need to be careful with the COVID vaccines. Now, the thing is, there are no adenovirus 5 based COVID vaccines, not approved ones anyway. There are some, I believe there's at least one in development, but there are no live ones anyway. So firstly, that's completely irrelevant. Then, at about the same time, the V-AIDS came out, and someone said, oh, if you have the booster, it gives you V-AIDS. So it depletes your immune system because of the booster. And also about the same time, there was another paper came out saying, we've identified a new HIV variant. Now, this HIV variant was actually first identified about 10 years ago, but it's only really just been sequenced and categorized as its own new type. So again, this was non-scientists reading a scientific paper and adding one and one together and getting 666. And all of those happened at about the same time, which just happened to be HIV Awareness Week, when the government every year, and has done for years and years and years, every year encourages at-risk groups to have an HIV test to know your status, which is a very, very sensible thing to do. If you are at a potential risk category for HIV, having regular testing means that you can catch the disease really early, you can get treatment, you can live an absolutely normal life, you can be non-infective, and if you catch it nice and early. So knowing your status is really, really important. But because these things happen all at the same time, they sort of get conflated together by social media. And there's that term, that a lie will be around the world before the truth's got its boots on. And there is a lot to that, because someone somewhere has looked at two of these things and stitched them together in their own mind, because they don't really understand the science behind it. And then, of course, someone else has stitched together one of the other factors, and then someone else adds on the other one. And all of a sudden, the rumor is there's HIV in the vaccines, and it's part of the depopulation plot. And it's utter nonsense, but it comes about from multiple misunderstandings all being conflated together into one thing.
Yeah, there was also a Australian, I think, vaccine.
Yes.
It didn't get past the first trial. Yes. Yeah. Sorry, go for it.
No, thank you for reminding me of that. I completely forgot. That also happened at the same time. And so there was an Australian vaccine, and it was a peptide vaccine. So it was just the spike protein itself. But the spike protein on its own is not really very stable. So what they did was they used a protein from another virus as like a clamp. Or if you imagine like an origami spike figure, it's folded together from paper, but it tends to fall apart because the two ends come apart. So all they've done is they put like a paper clip on one end to hold it in shape. But that paper clip happened to come from the HIV virus. Now, it can't give you HIV because it's just one bit of one protein from the HIV virus. But it did have a problem in the trials, which is what the trials are there for, because they thought, I wonder if it shows up on HIV testing. And it did. So they immediately stopped it after having vaccinated only a handful of people with it and abandoned it. But the BBC had made a program about it, and there was a short section where they talk about using this HIV peptide as a molecular clamp. And all of a sudden, that gets conflated with all of the other issues, and there's HIV in the vaccines, and yeah, it's going to kill everyone. And, zero truth, but what can you do?
Oh, this is it. Okay, I'm going to give you a couple of minutes, Graham, to plug your Immunology Explained.
Thank you very much.
And while you're plugging that, if anybody wants a question, if they DM me or Ivor or Brent.
Brilliant. So really, as a result of all of this misunderstanding, and immunology, as I said right at the very start, is incredibly complicated. It's very, very complex. I'm an immunologist. I've been an immunologist for 26 years. I know a lot about my specific field, viral immunology, but there's an awful lot of viral immunology I don't know. And there's a vast amount of other immunology that I've got a basic understanding of, a basic handling of, but it's an incredibly complex and broad subject, probably the most complex science there is. So there was a lot of people who wanted to know more about how the immune system actually works. What does it do? What does it do when you get an infection or you have a vaccine? And I thought, rather than go down the COVID route, I don't want to explain COVID itself. I want this to be more generic. So I started a Patreon called Immunology Explained. And that's all one word. You can find it fairly easily. And I'm posting.
I'll tweet it out later.
Yes, thank you very much for that. And on there, I'm posting a video every couple of weeks, although I'm going to do more if I can. And they're about 10 minutes each, and they explain in simple terms various immunological concepts. So chapter 1 of my posts, which I'm in the middle of now, is basic principles. So it might be adaptive versus innate immunity. It might be humoral versus cellular. It might be antibodies, which is what I'm going to record tomorrow. And then once we've done all the basics, I'm then going to build up, and I'm going to do a second chapter, which is going to be things like vaccine responses and primary versus secondary responses. And then we'll look at things like toll-like receptors and natural killer cells and some of the molecules. And to be honest, I can probably do this for years and years and years and still not run out of material because there is so much to learn. But what I've had very good feedback from people saying they've even sort of watched it with their teenage children who have understood it perfectly and have been captivated by it. And that's the thing. I want people to understand immunology, because if you've got a better understanding of immunology, then you're less likely to fall for some of the misinformation that is out there. So thank you for that little plug. That's very useful.
Okay. So I've got a question about sort of shingles and Bell's palsy after vaccination. And the question is, does the vaccination suppress the immune system transiently, or is it possible that it might cause some sort of autoimmune disease?
Right. So the Bell's palsy one is an interesting one. It's where the body attacks the ends of the nerves. And it's not particularly well understood, especially with vaccines, because in order to get that autoimmunity where the body attacks itself, you normally need some similarity between the protein you're vaccinating with and the target that the body will attack on the nerve or whatever. So I think actually it's a very different issue to shingles, which I'll come on to in a second.
Okay.
The Bell's palsy one seems to be, again, I think like the myocarditis, it seems to be more that person is predisposed to this sort of response. So you've almost got immune cells sitting there waiting to attack the nerves, and the vaccination boosts the whole immune system, and all of a sudden the nerve attacking cells are activated.
And Bell's palsy can happen after any infection, right?
Yeah, it can, and again, that's what makes people think that it is a predisposition. You know, much like some people have hay fever, and they've even found the potential for hay fever in people long before they're exposed to a particular pollen that kicks it off. So I think if you're predisposed to Bell's palsy, then again, like the myocarditis, having the infection is going to kick it off, as would the vaccine. So it's no consolation to those who do suffer that side effect. And obviously, you know, we would rather there were no side effects whatsoever. But it's not something that you're going to avoid necessarily by avoiding the vaccine completely. As for the shingles is a little bit different. So chickenpox is a very interesting virus. It infects nerve cells. And when you get shingles, you get a rash of spots on the skin. And interestingly, those spots are the end terminus of one nerve. So the nerve comes from your spine, it goes through the body, it gets to your back or your shoulder, wherever, and then it branches out like a tree. And that's where all of the branches end, in those places, because the chickenpox virus travels down the nerve, down each of the branches, and ends up at that location and forms a little spot there. Now, shingles is normally induced, or it's very often induced by stress. Again, it's not a particularly well-known trigger because, of course, trying to research it, you would actually have to monitor someone before the shingles kicks off. But people don't know it's going to kick off until they get it. So
yes,
it's really hard to monitor. But it seems to be kicked off by stress and things like that. So again, it's entirely possible that having the vaccination just kicks off some of the stress responses in the body and induces it. But again, the likelihood is, and I know that there have been cases of shingles after the infection. So the likelihood is it's the fact that you're generating an immune response that is causing these side effects, not the specificity, not the vaccine itself. It's the response to the vaccine, just as it would be a response to the infection.
If I could just add one thing very quickly, with Bell's palsy, it was noted that you get similar rates in general population. So that sort of lines up with your theory that you're always predisposed, but it's one thing what kicks it off. Yeah.
Yes. Yeah. Exactly. So unfortunately, it's one of those things where if you're going to get it, you're going to get it, and there's not a lot you can do. It's, in much the same way as people who in later life are going to develop arthritis. You're probably pre-programmed to that from birth, and it's no consolation if you've got bad arthritis to know that it's always been that way. But sometimes that is just what happens.
Yeah. Okay. I think I don't know if you've seen this. There was the Daily Mail printed something about a Moderna patent in 2016 about some cancer treatment or just cancer drug that they had. It had a similar sequence to SARS-CoV-2.
Yeah.
Thoughts?
Yeah. That one made me laugh. Now, I'm not as strong on the molecular side of things as I am on the cellular side, but it's basically what they've done is they've looked for a short section, and bear in mind they've got 19 amino acids that are the same, except that the full length of the spike protein, and I'm trying to remember how long it is, but it's a lot longer than that. Let's see if I can quickly find it. So it is, so it's 9. I mean, the full length mRNA, I think, is 9. Is it 9 kilobases, something like that? But you've got a long length. You've got the full length spike. Yes. Whereas, so the 19 amino acid or the 19 base pairs is a very short part of it. And in a way, it's a bit like opening a magazine and seeing a paragraph that you recognise from a magazine 3 years ago. Now, you might think, oh, well, they've copied it, or maybe the one 3 years ago knew that they were going to put, or whatever. But actually, that's really just doing mental gymnastics to try and make something fit your point of view. Because what you've got to remember is when Moderna made the mRNA that they did in 2016, I don't know off the top of my head what it's against. They're making it to target a biological molecule. So they're making it to target another virus, or they're making it to target to try and produce a biological protein. It might be a ligand, or it might be something else within a cell, or they want to knock something out, or whatever. So you've got in nature, you've only got a limited set of sequences that work. And if you remember back to sort of your A-level biology, and I was doing this the other week with my daughter, proteins, you've got alpha helices and beta sheets and so on and so forth. Well, these require a specific section of amino acids. A particular type, a particular sequence will be made to, say, put a bend in, or a hinge, or a binding site, or something like that. So those sort of sequences tend to be quite well conserved across nature. I mean, bear in mind, we share a decent percentage of our genome with bananas. Some of the proteins we produce, bananas produce too. So it's like saying, well, hang on, you've got a protein in bananas and they're around 10 million years ago, and humans have only been around 500,000 years. How did the bananas know that we were going to be around? Well, and so really, they made this sequence against a biological protein. So finding a very short bit with some similarity in another biological context is not really very surprising at all.
Okay, I'm going to make this my last question this evening. Okay. And it's a question actually that two people have asked about, which is there any evidence of a particular gene or set of genes that is associated with protection against SARS-CoV-2?
There are quite a few. So there are certain polymorphisms. That's small changes within the sequence of a particular protein. There are certain ACE2 versions out there that seem to be slightly protective. They don't seem to bind the viruses as well as others. And the one that is sort of more of my speciality is what's called MHC, major histocompatibility complex. And it was named because if you have a transplant, they look at whether you've got the same molecule sequences as the donor. And so that's why it's histocompatibility, tissue compatibility. And what this protein does in the body is it takes the bit of the spike protein and says to the body, this is what you're looking for. This is what the immune system needs to target. And everyone has different MHC types in their body. And they have found that certain MHC types present the viral proteins much more effectively to the immune system than other MHC types. And so although this won't protect you from infection like the different ACE types would do, it does mean that you can get a quicker and stronger immune response when you are infected or when you have the vaccine. Now, again, these changes might only be small. But you've got to take it into the context of all of the other different factors. So if it reduces your infection, how fast the virus replicates in your body by 10%, doesn't sound a lot. And if you get a huge, great dose of virus, then that 10% isn't going to make a lot of difference because the absolute amount of virus you're receiving is huge. But what it does mean is if you get a smaller dose and a milder infection after you've been vaccinated, then this helps you clear it that 10% quicker, which means that you have less severe symptoms, which means you may not even know you're infected. And a lot of the work on asymptomatic infection, so where people don't even know they've got it, is actually involved in looking at whole genome sequencing. Now, of course, that's complicated. We've got millions of genes, and each of those genes has got hundreds or thousands of variants. So trying to work, and also the big issue with asymptomatic is if you're recruiting people for an asymptomatic study, how do they know they've had it? Which they very often don't because they're asymptomatic. So it's a difficult thing to study, but there are quite a few papers out there showing that various proteins do have specific changes.
Okay, I'll be honest. Whenever I said that was my last question, I lied. I got another one came in that I think we should ask every guest.
Okay.
And then I'm going to So, which is the single piece of COVID-related disinformation that you think has been the most damaging?
Overall, I think it's the pregnancy one.
Okay.
So Yeadon, who of course claimed to be CEO of Pfizer, or his followers did, and so on and so forth, came out with a claim relatively early on that the spike protein shared homology. That's to say it had similar sequences or the same sequence as something called syncytin, which is found in the placenta. Now, as a result of that bit of misinformation, and because of his background working for Pfizer and so on, it spread rapidly. And talking to various sort of obstetricians and midwives and so on and so forth, there was quite a significant reduction in the number of pregnant women coming forward for their vaccine. And it had been proved to be safe. It had been tested and it had been shown absolutely safe and very, very beneficial for both the mother and the baby. But a huge number of women refused the vaccine based upon this misinformation. And anecdotally, at least, there were deaths of both mothers and babies from COVID when there didn't need to be. And it's really frustrating because it was such an obviously incorrect claim, because the homology showed was 6 base pairs long. Now, I remember someone at the time, they did a search in the human genome for those 6 base pairs, and it cropped up in something like 15,000 different proteins. It cropped up in several proteins in goldfish and so on.
Yes.
So if it occurs in now, and it also ignores the obvious point that if the spike protein is similar to syncytin and you make antibodies against it and therefore attack the placenta, if you become infected, you're going to make some antibodies at least against the spike protein, and you're going to attack the placenta. So it rather ignored the point that infection is going to do the same as the vaccine would if it were true, which it's not.
Yeah, this is the thing that to me as a layman, it seemed obvious. Well, it seemed to me that if this was true, we had to absolutely go completely zero COVID, literally army on the street,
But of course, at the same time, people like Yeadon were complaining about lockdowns and masks.
Yeah, it made no sense.
So it made no sense at all. And from an immunology point of view, it didn't make any sense anyway, because even if it did have homology, the body doesn't see, the immune system doesn't see chunks that are only 6 amino acids long. It sees chunks that are 10 to 11 amino acids long. And if you expand this homology said out to 10 or 11 amino acids, it doesn't line up with anything on syncytin. And it also ignores the other immunology point, whereby normally, if you have a cell that will attack your own body, it gets deleted before it can become a mature cell. So you educate your immune cells so they don't attack your own body. That's why we don't attack our own bodies constantly. So even if there was homology there, in the vast majority of people, you would filter it out anyway. So the whole claim could be debunked in about 2 or 3 minutes, but it caused deaths. It caused deaths of babies, and it caused deaths of pregnant mothers and new mothers. And that, to me, is completely unforgivable.
Yeah,
I will actually say great minds do think alike, because we asked Dr. Dan Wilson the same question. I assume you know who he is.
Yes,
and he gave the same answer, yeah.
Yeah,
but he gave it from another perspective.
Yeah, well, again, there are different points of view, and another person you could have onto a future one is Vicky, who and I'm going to give her full name, who works on pregnancy and reproductive immunology, and she is absolutely fantastic, and she's sort of debunked an awful lot in that specific field, and it's so frustrating when something is blatantly false, is being spread around that way, and then you see, even on social media, you see a pregnant woman saying, well, I'm pregnant and I'm not having it because it'll kill my baby, and part of you just hopes and prays that there's not a disaster from that, and that she doesn't regret that for the rest of her life.
Yeah.
But it is a major part of the problem that things like that sound believable to a layperson, and if you've got the ex-CEO of Pfizer, as you're told, telling you this happens, as a layperson, you probably believe it.
Yeah. So that was Vicky Meal you suggested, right?
Yes, that's it. Yeah.
Yeah. Okay. Okay. Well, like I'm out of questions for now, Graham. Thank you so much.
It's been fun.
And I don't know if Brent or Ivor have anything to say, but I think that was great. You dealt with questions which were probably difficult, which were probably unexpected, really well, and I hope it was useful for people. We'll make sure that people can download it and listen to it.
And the interesting, the thing I'll sort of finish with is, you often get anti-vax saying, I've done my own research, which tends to be looking at Facebook or BitChute or something like that, for a video. What I would say is, it is good to encourage people to do their own research, read around, but read reliable sources, read people or watch people who can back up what they're saying with facts, with evidence, with explanations, and more importantly, who are prepared to defend that view. Because if you do your own research, if I turn around to an anti-vaxxer and say, no, you're wrong, why should they believe anything I say? But if I say, no, you're wrong, look, here's how it works, here's 4 or 5 papers proving it, then they may well turn around and say, actually, you're right. And then, maybe disaster averted there in the future. You never know.
Yeah, absolutely. Well, look, thank you very much.
Thank you for asking me. It was really good fun.
Great.
It definitely was, yeah.
Yeah, thanks for coming on. It's brilliant.
Brilliant. No, thank you.
So I guess we should announce our next guest then. We will be talking to Linda Green from Back to the Vax whenever we have that arranged. So she is a Canadian lady, so I dread to think what the poster's going to look like for her, who used to swim in the anti-vax waters and has managed to get out, and she is now encouraging people to get vaccinated. Generally, she's doing this, I think, from before COVID. So I think it'll be a very interesting discussion on the anti-vax movement.
I believe she got out of the movement just before COVID hit, actually. So it would be really interesting to get that perspective, because obviously with me and Brent as like ex 9/11 truthers, to sort of like how does it interact with science in the same way, and what is the psychology of it, I think they would be very similar.
Yeah, absolutely. So I guess so I guess we'll hear from you all again, and I hope you all have a lovely rest of your evening. Thank you very much, and good night.
Bye.
Thank you.
Bye.
Bye.
Bye.